So chapter 28 is on the nervous system. The nervous system receives sensory input. So it receives stuff from the outside world and then allows us to interpret that and then send out an appropriate response to that. So we can take information that comes to us, analyze it, and then do something about it. It is the most intricately organized data processing system on Earth. Humans have a tendency to make the brain whatever, an analogy of the brain, whatever is the newest, coolest thing. So when machines during the Industrial Revolution, machines were becoming really popular, biologists would say, well, the brain is just like a machine. And now computers are in, and we say, oh, the brain is just like a computer. It's actually more intricate than that even. We can't really replicate it artificially yet. Certainly they're working on that. They're really trying, but it's a very intricate system. Okay, so the nervous system obtains and processes sensory information, whether that is touch, feel, smell, sound, taste, or whatever. It's going to take that as an input, analyze it, and then do something about it. All right, the sensory neurons conduct signals from sensory receptors to the central nervous system. So you have sensors all over your body that are going to sense different things. Temperature, heat sensors, you have touch sensors, you have all kinds of different sensory receptors. Taste, all those things, smell, and that goes to the central nervous system. The central nervous system, what we define that as, is the brain and the spinal cord. That is the central part of it. And that is going to do the analyzing of the input. There are interneurons in the central nervous system, and they integrate information and send out a response to the motor neurons. The motor neurons then convey a signal to whatever effector cell needs to be activated or affected. Okay, so you have automatic responses when you go to the doctor and they whack your knee with the little hammer. That actually doesn't go to your brain, so you can't really control your response to that. It goes to your spinal cord. So the sensory neuron here is going to feel the stretch, right? We're going to stretch this tendon. Whacking this causes this to stretch, okay? A ganglion is just a bunch of cell bodies, right? That's going to go to your spinal cord. Your brain doesn't get to analyze this. The spinal cord is the central nervous system part that's analyzing this. And you get a reflex response, which is to kick the doctor. All right? So it doesn't go to the brain, but it does go to the spinal cord. All right, located outside of the central nervous system is the peripheral. Peripheral just is outside of the central, right? Nervous system, and it's going to consist of nerves, bundles of fiber of sensory and motor neurons, ganglia, that's the bodies, clusters of cell bodies of the neurons, right? Okay. Neurons are the functional unit of the nervous system. That's your cell. That's the cell body. They're very, very specialized for their job. They have one job, sending and receiving information, and they do that very, very well. So their function really affects the structure of it. If you look at this, the cell body, you have the body, but you have all these freeways where we could send or receive a signal along. Dendrites from the word tree, because think of this as branching out everywhere. Okay? So you have a direction, you're going to have some kind of input. So here we have an input to the cell. It's going to go down, and these little nodes of Ranvier are, they help the signal travel faster. It helps the signal kind of skip from node to node to node, and that increases the speed of the signal. And it's going to end up, so it goes from the body right down the axon. These little nodes speed it up. We'll get to these little Schwann cells in a minute. They are support cells. It's a myelin sheath. They make a sheath around this. Again, it increases the speed of the signal. But the other thing this little sheath does is prevent sort of short-circuiting of the signal. You wouldn't want the signal coming down here and then jumping out to another nerve nearby. We want to keep it one directional so we're not spazzing out all the time. And then it goes down to the synaptic terminals, and this could be another nerve here that is going to respond, and we're going to send the signal from nerve to nerve. You can send it down to your effector cells. You can send the signal, so wherever it's going, it's this direction, comes to the dendrites, down the axon, to the synaptic terminals, and off to do whatever it needs to do. Okay. Many axons are enclosed by cellular insulation called the myelin sheath, and that's what we saw on the previous slide. It speeds up the signal transmission because it causes the signal to jump from node to node, and it also prevents sort of this short-circuiting effect where it could jump out of the axon and randomly stimulate a nearby cell by accident. A neuron maintains a membrane potential across its membrane, and this is called the resting potential. And the reason it's this way, so we have literally an electric voltage as the neuron is at rest. And this is involved in potassium and sodium ions being pumped across this membrane here. And it's about a 10 millivolt difference. But the real efficiency of the system is that it stays charged while it's at rest. So when activated, these ions can quickly switch places and send a signal by having this resting charge, as opposed to if you had a signal coming and you had to try to make a charge by pumping ions. That would just take forever. These signals have to go very, very quickly, in milliseconds, go through a nerve. So by having a resting charge as it sits there, a resting potential, it could send the signal much, much faster. Okay. The resting potential is caused by the membrane's ability to maintain a positive charge on its outer surface and a negative charge in the inner surface. And this sodium-potassium pump is key to this. So it is going to actively pump and create this charge, right? It's going to actively pump out your sodium ions and pump in the potassium ions. You're going to get active pumping of ions to create a resting potential. Then we have these channels that will allow free movement of ions. They're going to actually open when the signal is sent, because that's a much faster channel than this sitting here and pumping it. So this is involved in creating that resting potential. Okay. The nerve signal, when we activate the nerve, begins as a change in the membrane potential. A stimulus alters the permeability of a portion of the membrane. So if we alter that permeability, remember we're going to let ions cross the membrane, you're going to get a change in that millivolts, the potential, the electric potential. It's like, think of an electric charge, literally. You're going to get a change in ions, and that is going to travel down. It's going to pass that change, that signal down the nerve, down the axon. Okay. The nerve signal, it's called an action potential. So we have our resting potential, and then we have an action potential. And it's the change in the membrane voltage, literal voltage here, from the resting potential to a maximum level and back to the resting potential. So as we look at our little diagram here, look here. You have sort of this resting potential. Here is a neuron just hanging out at rest. You have what's called a threshold, okay? You have to meet that threshold requirement. Your cell can't fire every single time an ion passes through the channel. You'd be spazzing out all day long. You have to meet a threshold requirement where we've had enough change in potential that the channels will open. The ion channels will open. You'll have a spike to what's called the action potential. This is the nerve signal. This is going to be a spike. You have a slight, as it returns to the resting potential, it kind of overshoots the mark a little bit because these are ions going, in and out of the membrane. It tends to overshoot a little bit, and then it goes back to the original resting potential. And that will propagate down the cell. This opening of the membrane channels will propagate down that axon, and the electron potential will literally go, not like a wave, but kind of jump from node to node down the axon. Okay? The action potential propagates itself along the neuron, and the action potentials are self-propagated in a one-way channel, so one direction along the neuron. It's an all or none event. You have to trigger the nerve. You have to meet that threshold requirement to create a trigger effect. And once you've done that, the channel's open, the ions flow through, and you have the action potential. All right, so let's look at the propagation of the action potential along the axon. What you have here is you're going to have sodium coming in, rushing in to the cell. This is the action potential right here. So we're going to look at this signal traveling down this axon, okay? So here we are, we're right here, sodium rushes in. That's going to cause the membrane next to it to open these channels, and sodium's going to rush in here. Meanwhile, this area is going to return to the resting potential. How does that happen? The potassium has to come out, all right? It's going to return. So the action potential is now here, and that's going to trigger it to move to the right. So it's going to come here. This is going to return itself to the action, to the resting potential, excuse me. Remember, it's going to overshoot a little with the potassium here. And so it propagates down from left to right in this one. It's always one direction. Okay. So then the neurons talk to each other at what are called synapses, which is a little gap between the neurons, or between the neuron and whatever cell they're trying to affect. It's a little junction, a little gap, or whatever you want to call it, between the cells. Okay. So the electric signal passes between the cells of electrical synapses, and then you have a chemical synapse that are secreted, and that's a chemical signal, and that is called a neurotransmitter. And you might wonder why our nervous system would want to use both electrical action potentials and chemicals. Why are we doing the mixing? And the thing with these neurotransmitters is they can do a variety of things. So so when we activate them, some of them can make you sleepier, or some of them can make you more excited. The reuptake of them, when they get reabsorbed, you can regulate that. So you can do a variety of things using the chemical synapses. So I think that's probably why our body has sort of both versions of this. The neurotransmitter crosses the synaptic cleft and binds to the receptor of the surface of the receiving cell. So it's going to cross that little gap and go to the other cell, physically go across the gap. Okay? So here's a nice picture of it. You have inside the nervous cell, the nerve sending the cell, you have these little vesicles that are going to contain different neurotransmitters, right? And it's going to depend on which one, what signal. We're going to literally fuse the vesicle here to the cell membrane wall. It's going to pop open, kind of like a little zit. It's going to allow the neurotransmitter to come and bind to the receptors. All right, and then these are going to do different things. With ion channel, it's going to let things in. And then these are going to get reuptake. They're going to go back to the cell. They don't hang out in this synaptic cleft forever, because then they would keep firing this next cell, right? So it's going to be a temporary thing. They're going to come out. They're going to do what they need to do in the synaptic cleft here, and they're going to get reabsorbed. The neurotransmitter binds physically, right? The physical binding can be to the ion channel and cause it to open up, allow ions to flow in and out of the receiving cell, and then it's released back into the cleft and ultimately reabsorbed. Chemical synapses make complex information processing possible. That's why I said, why have chemicals? Why not do everything electrically? Well, because the chemicals can do different things depending on what they are. Okay. And when we look at this neuron, a neuron can receive information from hundreds of other neurons. Okay? So it literally, if you look at this, it has little kind of receivers all over itself. If you look, there's ones that, all the green ones here are what are called excitatory. that will excite this one, and there's red ones which are called inhibitory, so that it won't send a signal, right? So that we can activate parts of our brain and not having all of them fire. So, a signal going one way that's going to excite one cell may inhibit the next cell, so that things can be very directional and controlled. And down here you can see an actual picture of all the synaptic terminals on this thing. So it can take a lot of information into it. Okay. Some neurotransmitters excite the receiving cell. Other neurotransmitters inhibit the receiving cell's activity. So it can do that by decreasing its ability to develop action potentials. So again, this controls things, right? We can excite some things and then not spaz out again and have everything firing at the same time. We want controlled information flow. Okay. The peripheral nervous system is a function of hierarchy, all right? So it's divided into two functional components. You have the somatic nervous system, that's your body, and then you have the autonomic. And think of that as automatic. Like the autonomic does things without your control. You don't really have to think about them. Good examples of that is you're digesting your food right now and you don't have to think about it. You're secreting hormones right now. You don't have to think about that. You're breathing right now. You don't have to think about that unless I tell you to. Your heart is beating. You don't have to think about that. And so that lets your brain focus on things you want to think about. Whatever movement you're doing, you're taking notes, you're writing, you're thinking about your hand, you're thinking about the lecture. And so the rest of your body can just kind of go on with life without you having to concentrate on it. Okay. The somatic nervous system carries signals to and from skeletal muscles, mainly in response to external stimuli. So this is mostly our response to the outside world. The autonomic nervous system regulates the internal environment by controlling smooth and cardiac muscles and the organs of various body systems. So again, I can sit here drinking my Diet Coke and my body can digest that, my liver can detoxify that, and I don't have to think about it. I can think about doing the lecture or talking or moving the mouse or whatever I need to think about. Opposing actions of sympathetic and parasympathetic neurons regulate the internal environment. So the parasympathetic division of the autonomic nervous system primes the bodies for activities that gain and conserve energy for the body. So I like to think of the parasympathetic nervous system as sort of the food coma nervous system. This is after you've had Thanksgiving dinner and your pupils are constricted, they're not dilated, you're not expecting a tiger to come out and eat you, you're kind of sitting there fat and happy, you're very relaxed, as opposed to the sympathetic part of the nervous system, and that's going to be that adrenaline rush where your eyes dilate and your heart rate increases and you get all excited and you're ready to run away from the tiger. So those two are opposite of each other. Okay. So here you have examples of the parasympathetic nervous system, and your pupils, like I said, are constricted. You've stimulated your salivary glands. You are, digesting your Thanksgiving dinner. Your lungs, you're not requiring extra oxygen right now. You're just kind of chilling out. Your heart rate's low. We are digesting food probably because the whole point of this is to kind of hang out and digest. This is also why I don't really suggest that really, really, really big breakfast right before an SAT. Might make you need to go pee because you're not really running from a tiger anyway. And it can promote an erection of the genitalia. So when you're just kind of sitting there, you have blood flow to all different parts of your body, including your genitals. Then you have the sympathetic division, sort of the exact opposite. You now have a tiger coming at you, or a biology test. Your pupils will dilate. Your mouth can get really dry, especially if you get really nervous about your biology test. You breathe in more air. You need more oxygen. Your heart's beating faster to move that oxygen around. The gland stimulating are going to release epinephrine and norepinephrine. Um, you're going to release glucose into your bloodstream because your body is assuming you need the sugar to go move your muscles or do whatever you need to do. You are going to stop digesting. So, whenever you eat something and then either really stress out or go work out, that food feels like it sits in your stomach for a long time because it does sit in your stomach for a long time. You're not going to have to go pee because you're too busy doing other things. And then sexually, it would promote the ejaculation and vaginal contractions, like an orgasm. So when we look at sort of sex ed, it'll involve the parasympathetic and the sympathetic